Hi, this talk is part of a symposium on studying the individual, and here I'm going to focus on how time-resolved internal states can be valuable for studying individual differences and in developing markers of health and disease. First, I think it's interesting to think about fMRI scans as showing us something like a movie of how activity is changing across the whole brain. And there's been more and more research focused on dissecting these kinds of rich fine-scale dynamics and using that information to learn more about individual people. So toward this goal, here I'm going to discuss two directions that we've been studying in our recent work. One involves internal states, and so the way a person's brain activity looks across different states, such as wakefulness, emotion, and so forth, that may differ between people and may be altered in disease, and so might the dynamic transitions between these states. A second idea, which is not unrelated, involves brain-body interaction. So many imaging studies focus only on the brain, and we tend to treat physiological effects as noise or ignore them, but there's growing evidence that integrating brain and body information can also give us new insight into individuals. So on the first topic, there's been a lot of exciting work from many different groups that's showing how time-resolved functional brain states can be detected from fMRI signals, even across short windows of time. So for example, functional connectivity patterns within some period of time can indicate things like levels of sustained attention, changes in cognitive state, and mind wandering. So in this direction, one state that we've been focusing on is arousal, which captures a spectrum from being very alert and wakeful to drowsy and to being asleep. And one reason that this is important for fMRI studies is that different arousal levels come with differences in brain activity as well as behavior. And I think that especially as we move towards studying individuals rather than group level averages, this becomes quite important since whichever state a person is in during their scan will be reflected in the fMRI patterns that we're using to represent that person. However, I don't think we should just consider this to be something like a confound. So one thing I actually wanted to highlight in this talk is that the fMRI spatial signatures and temporal dynamics of arousal may themselves provide useful markers of individuals and disease. And so some of the questions we've been asking along these directions are how are arousal levels expressed in fMRI patterns measured across the brain? And are these patterns sensitive and robust enough to actually detect how alert or drowsy somebody is at any given moment in time? So in this study, we examined if arousal levels can be detected in the low-dimensional topology of fMRI data. So for example, if we take the fMRI activity pattern at a single TR, divide that into ROIs, and project that into a lower dimensional space, for example, 3 dimensions, which is something we can visualize, would the resulting organization of these patterns in low dimensions show any relation to arousal? And so this was the result from a particular data set where subjects were performing a reaction time task. Each point is an fMRI volume at 1 TR that's projected into 3 dimensions, and we colored that point according to how quickly or slowly somebody is responding around that time. And there does seem to be somewhat of a gradient where more drowsy or more alert time points seem to cluster in similar regions in low dimensions. We had also recorded EEG signals at the same time, and we find that if we were to instead color those same data points according to an EEG spectral arousal measure of arousal, then we also see good agreement. So if we cluster these low-dimensional points into discrete clusters and look at the cluster membership over time, which is indicated by the colors here, we can see that the time points that fall into these distinct clusters seem to align quite well with external measures of arousal, which in this case was EEG since it's a resting state scan. And one thing to note is that this clustering was done in a fully unsupervised way, so we didn't input any information about arousal to derive these clusters. It was just the fMRI data in a low-dimensional space. And so when we divide the data into this this is if we divide the data into 2 clusters, but if we allow for larger numbers of clusters, we saw that they interestingly seem to nest themselves in within these coarser arousal bins in many of the subjects. So that may further indicate that different arousal levels come with distinct fMRI activity patterns. So what are the activity patterns that are happening during these time points? If we plot the average whole brain activity from each cluster, we saw that in the clusters corresponding to higher EEG-defined arousal levels, there tended to be clearer network structure and a lot of anticorrelations across the cortex, whereas for those in clusters corresponding to times when EEG indicated the subject may be more sleepy or asleep, this seemed to be more uniform across the brain in terms of the sign of activity being mostly positive or mostly negative, although some network structure is also apparent as well. One thing I wanted to point out is that we see quite a dramatic alignment with arousal in this particular scan, but we may not expect to see that for any particular scan. In this study, the data that we used involved long resting state scans with eyes closed. Subjects are more likely to fall asleep, as well as sensory tasks with very sparsely presented stimuli, so it's also somewhat of a boring task where these changes in arousal level may be more prominent. Overall, though, this study highlights how studying internal states like arousal may be detectable in fMRI data throughout individual scans and possibly in individual time points. And it also suggests that parsing fMRI scans by vigilance levels and looking at the corresponding patterns, we may see different patterns, and these might be useful for understanding individual differences. In addition, we can also ask whether fMRI scans can track more continuous variations in someone's arousal level across the scan. So in other words, if we're given an fMRI data set as input, can we output a one-dimensional time course that is an indicator of how alert somebody is across the scan? So here we use a supervised learning approach. We use simultaneously recorded EEG signals in order to learn a pattern of fMRI data that directly tracks increases and decreases in an EEG spectral measure of arousal. And then the idea is that since most data don't have concurrent EEG, we'd be able to take that pattern, that previously trained pattern, bring it to a new data set that has only fMRI, and if we project that along the fMRI timeframes in that new data set, then we can derive this time course of estimated arousal level, which is shown in red. So we show an EEG measure for comparison here, but we did not use that to derive the measure in red. We only superimpose it for validation. So one thing we wondered is, in addition to tracking EEG, can this fMRI arousal metric also predict variability in a person's behavior? And so to test this, we had subjects do this simple sensory task that presented auditory tones at a sparse and random fashion, and subjects were asked to make a button press response as soon as they heard the tone. So here we looked in a 5-second pre-stimulus interval before each tone was presented, and we asked if our fMRI alertness estimate could predict that subject's response to that tone. And what we find is that for most individual participants, the level of alertness that we estimated from that fMRI measure was higher right before the trials where subjects did respond to the tone compared to when they missed. So each one of these connected dot pairs is a subject. And if we zoom into a chunk of time around the presentation of each stimulus and we show the average time course of arousal estimate for the trials where subjects responded compared to the ones where they missed, we can see that these measures do diverge even before the onset of the stimulus. If we then only take the trials where a subject does respond, we wanted to see if it is higher before the trials where they had faster responses compared to slower responses, which also seemed to be the case for many of the participants. And here's the results when we pool the trials across all the subjects and divide those into fast, slow responses and misses. So together, these studies show that fMRI may be sensitive enough to track changes in arousal over time. arousal information may be detectable in the low-dimensional structure of fMRI, and these fMRI-based measures may be predictive of subjects' behavioral responses to ongoing stimuli. So our current methods are published and publicly available, and we are also making extensions based on deep learning in order to further improve the extraction of EEG arousal measures from fMRI. We hope that this can enrich the study of individual differences in at least a few ways. So one is, if we are able to model arousal very well, then this may help to separate those effects from other kinds of brain processes instead of just leaving those all mixed up in someone's data. So that can help to increase the precision and the interpretability of the fMRI scan. But second, we may also be able to use arousal information itself as a marker of interest. So as one example, Kate is an undergrad in our lab who will be presenting work here at OHBM, where she's looking at how fMRI patterns linked with arousal are different in patients with mild cognitive impairment, and how those patterns may also relate to inter-individual differences in cognition. Hatef is a PhD student in our lab who has been studying functional connectivity of subcortical nuclei that are implicated in arousal and autonomic function. And one of the things that he's been finding is that in patients with epilepsy, there's reduced connectivity from these structures across a number of brain areas, even when we match vigilant states between the patients and controls. So moving to the next topic, another interesting and related aspect of internal states involves autonomic physiological processes in our body, such as breathing and heart rate. And there's growing evidence that studying the brain and the body together can greatly enhance our understanding of individuals. For instance, it may reveal trajectories of aging and disease, and changes in physiology may be closely tied to brain function. So here it was found that daily training in increasing the variability of your heart rate may be beneficial to emotion regulation networks in the brain. A second reason to study physiology is that fMRI signals, of course, are based on blood oxygenation, and so changes in breathing, heart rate, and other physiological variables can directly change fMRI signals and connectivity patterns in the brain. And so if we don't account for this, then it may obscure other kinds of neural interactions. I also wanted to highlight a couple of the symposia that are happening at this meeting on these topics, which look really exciting. So physiological signals from the body can be very valuable to have during our scans, but in practice, there are often barriers to studying them. So firstly, many fMRI data sets either haven't recorded physio signals like breathing and heart rate, and even when they are recorded, we often find that the quality isn't sufficiently high, and it can be laborious to look through all these recordings and try to correct, errors and artifacts in them. And so to overcome these barriers, we and others have been working on methods for trying to extract physiological information directly from fMRI scans. So in this work led by Rosa in our lab, we've been developing deep learning models to extract low-frequency fluctuations in the volume of your respiration, as well as your heart rate across the scan. So in other words, given fMRI signals from the brain, can we reconstruct the dynamic changes in somebody's breathing and heart rate during that scan? Here we're using data from the Human Connectome project, and to show a couple of the results, these are two example predictions of respiratory volume time courses and heart rate signals that we derived from the resting state brain fMRI data. And each one of the predicted waveforms here is overlaid on our ground truth recorded signals since the sensors were available. So as you can see, these are examples from very successful runs, and here's how it performs across all the held out scans. We measured our performance based on the Pearson correlation, temporal correlation between the predicted and the measured physiological waveforms. So those findings are in the HCP resting data set, but we've also been exploring the direct generalization to other task conditions. So here's looking at several of the HCP task data sets. And we're also looking at data sets with very different acquisition parameters. So in this one, the TR was actually 3 times slower than the one in Human Connectome project data. We've also been trying to extend these models to work well across the human lifespan, and so, in aging participants, and ultimately are hoping to make these tools for studying physiology that are powerful and that generalize well over a range of data sets. So how can this help us to study the individual? As a couple of illustrations, here we found that if we remove the physiologically induced component of fMRI signals based on our model prediction, it has comparable effects to when we removed a component based on the measured physiological signals. Oops, I had those reversed. And second, that's kind of thinking about physiology as more of a confound, but we've also been looking at whether this tells us something useful about people. And so in this study, we were finding that the patterns across the brain of fMRI signals that correlate with physiological variables, such as breathing and heart rate, these seem to be different between people. They may be also stable within individuals over time, relatively speaking, and may correlate to cognitive variations. So lastly, I wanted to note that although I presented arousal and physiology here separately, there are also close interconnections between these processes. For example, major components of spatiotemporal variance in fMRI signals appear to correlate with multiple arousal and physiological signals, underscoring this unification of the brain and body. And here, Taylor Bolt is going to be presenting on this collaborative study that he has led. So in summary, dynamic arousal and physiological states may be important dimensions of individual variability. We may be able to extract these dynamics and states from fMRI data itself, even in individual scans. Our ability to unravel these components from other signal sources and effects in fMRI can help to advance the overall precision of fMRI for describing individuals. And while many studies are considering both arousal and physiology only as confounds, there's increasing evidence that points to their potential as interesting markers of an individual. So thanks for your time, and thanks to my lab and collaborators.